Welcome back everyone. In this video we're going to be looking at active transport in cells. Now this image, in case anyone's into Greek mythology, shows Sisyphus, who was an ancient Greek king according to Roman Greek myth, and he was condemned by Zeus to forever roll a ball up a hill and never get to the top. That takes a lot of energy, and he is definitely going against the grain. Okay, so for this lesson we're going to be looking again at the keyword selectively permeable with regard to membranes. It's about active transport and adenosine triphosphate, or ATP. We're looking at the concentration gradient, carrier proteins, vesicular or mass transport, and of mass transport that can be classed as endocytosis or exocytosis, and types of those are pinocytosis or phagocytosis. We are going to be focusing on the remaining learning objectives that will capture those types of transport, and again we're going to be looking at how the concentration gradient can influence this. Okay, so what is active transport? It is actually the movement of substances against the concentration gradient. So you're going up instead of down, hence the picture of Sisyphus. Molecules are traveling from high to low concentration, and they need carrier proteins to do that, and this requires ATP as an energy source. So very different to passive transport. In this diagram here, you've got an illustration of that. So you've got a small number of molecules here, but they're actively moving into the cell. So they're being pumped in through this protein. And this process is consuming energy in the form of ATP. So again, here we're talking about energy being required, and we're going against the concentration gradient. Again, short video just to summarize what active transport is.
Cells must also pump molecules against the concentration gradient. Ions such as sodium and potassium are moved in this way. Again, a special carrier protein, in this case the sodium-potassium pump, is involved. ATP transfers energy to the carrier protein. This causes the carrier protein to change shape so that sodium ions readily bind to it. The binding of sodium causes another shape change, and the sodium ions are released outside the cell. The protein can now bind potassium ions. When the potassium ions bind, the protein once again changes shape, and the potassium ions are released inside the cell. At this point, the energy gained from ATP has been used up. The sodium-potassium pump needs energy from another ATP to repeat the cycle. The pump moves sodium ions against their concentration gradient. It also moves potassium ions against their concentration gradient. Moving ions this way requires energy, and thus is an example of active transport.
Okay, so at this point we should have a really firm understanding of passive transport and active transport. So I'd like you to stop the video here and see if you can compare these two types of transport. So for comparison, you should have at least 2 similarities and 2 differences. See if you can get 3. Okay, hopefully you were able to do that, and just as an example of how you might lay out a question like that, we've got active transport and passive transport, and I have got 3 points of comparison here. Now, a really big tip for when you are writing answers in a test or an exam, whenever you see the word compare or draw a comparison, draw a table. It doesn't matter if there are lines there, draw a table over that. It's a really useful way of checking that you've got all the marks, and it's really easy for your marker to see that you've understood the question, and they can mark that easily and not miss out on anything. So this is a really good habit to get into early on. Please make sure that you make an attempt to draw tables for compare questions. Okay, so the next type of mass transport we're going to be looking at, or active transport, is vesicular transport. So as the name suggests, we're looking at transport of molecules in vesicles. Now this is described as mass transport sometimes because we're moving quite a lot of material in the one go. Again, this requires energy because you're moving lots of stuff in one go, so it's a form of active transport, and it can take things into a cell, so it's endocytosis. So endocytosis is taking vesicles containing liquids or solids into a cell, or it could be exocytosis. So this is when the contents of a vesicle are passed out of the cell. So the word exo means out or to leave in Greek, which is why we use these terms. So again, another short video describing endo and exocytosis for you. Typically, endocytosis, we might use something like bacteria, and for exocytosis, if we want something to leave the cell, that would be secreted out.
Cells can move large particles across the membrane by bulk transport. Bulk transport into the cell is called endocytosis. Bulk transport out of the cell is called exocytosis. During endocytosis, the cell membrane folds into a pouch surrounding particles outside the cell. A vesicle is formed. This vesicle carries the particles into the cell and can release its contents into the cytoplasm. Human white blood cells engulf bacteria and other unwanted cells. This process is called phagocytosis. Phagocytosis is another example of endocytosis. Once inside the cell, the vesicle containing the bacteria fuses with another vesicle called a lysosome. The lysosome contains digestive juices which destroy the bacteria. Exocytosis is the opposite of endocytosis. Particles are transported out of the cell. A vesicle containing wastes or cell products moves toward the cell membrane. The membrane surrounding the vesicle fuses with the cell membrane. The contents of the vesicle are secreted as the membrane smooths out.
Okay, so now we have an understanding of endo and exocytosis. We're going to look at phagocytosis. So again, this is a form of endocytosis, and it literally means cell eating. So this is when solid particles, hence eating, solid particles are taken into the cell by the cell membrane folding around it, forming that vacuole. So that's what we looked at in the video there. Typically, it would be a bacterium that the white blood cell wants to ingest. So once we've had the membrane folding around this solid particle, it traps it in a vacuole, and that vacuole moves away from the cell membrane and it moves into the cytoplasm. Pinocytosis, slightly different. It doesn't mean cell eating, it means cell drinking. So similar process, but with liquids. So again, liquid particles are trapped in a vacuole by the membrane folding in on itself and nipping off to form these vesicles, which will then migrate away from the cell membrane and towards the inner part of the cell. Okay, that's the end of this lesson. I'd like you to please make sure that your booklet is completed up to and including page 22.